Picture a cone snail, a small, beautifully patterned shell drifting across a tropical reef. Slow, soft-bodied, seemingly harmless. It doesn't look like a predator. It doesn't move like one. And yet, hidden inside that shell is something that can kill a grown human in under an hour. A venom so precisely engineered that one compound derived from it is considered 1,000 times more powerful than morphine. Now picture a platypus, the duck-billed, beaver-tailed mammal that looks like evolution assembled it from leftover parts. It's venomous too, not through fangs or stingers, but through spurs on its hind legs, producing a venom that causes excruciating pain that no known painkiller can touch. Here is what shocked me when I started digging into this. Those two animals share no meaningful common ancestor. They are separated by hundreds of millions of years of evolution. They live in completely different environments. They hunt completely different prey. And yet both arrived at the same solution: venom. This isn't a coincidence. It's a pattern. And once you see it, you can't unsee it. Across the animal kingdom, venom has evolved independently on more than 100 separate occasions, not variations of the same ancient system passed down through generations. Entirely new systems, built from scratch, in creatures as different as jellyfish and shrews, spiders and fish, snakes and slow lorises. Each time, evolution looked at the same problem: how to subdue prey or deter a predator without a prolonged struggle, and kept arriving at the same answer. Scientists call this convergent evolution, the idea that similar pressures produce similar solutions, even in unrelated lineages. We see it in wings, which evolved independently in birds, bats, and insects. We see it in eyes, which evolved over 40 separate times. But venom might be the most striking example of all, because it isn't just the concept that keeps reappearing. It's the chemistry. Consider phospholipase A2, an enzyme that tears apart cell membranes. It shows up in bee venom, in snake venom, in scorpion venom. These animals diverged from one another so long ago that they barely share a biological blueprint, and yet they all recruited the same molecular weapon. Or another example would be calcium channel blockers, compounds that interfere with nerve signaling, independently evolved by snakes, spiders, and cone snails. The same biochemical target, reached by completely different paths. This keeps happening, over and over. Life reaches into its molecular toolkit and pulls out the same solution. To understand why venom keeps reinventing itself, you have to understand what it actually is, and what it isn't. Venom didn't evolve as a killing machine. That's not where it started. In spiders, as we've explored before, the earliest venom was likely digestive enzymes that migrated into glands near the fangs. In snakes, the story is similar. Proteins already present in the body got recruited into new roles. Saliva proteins that had spent millions of years breaking down food suddenly found themselves being expressed in higher concentrations. This process, what scientists call toxin recruitment, is the engine behind venom's relentless reinvention. Evolution doesn't build from scratch. It tinkers. It takes a protein already doing one job and figures out how to make it do another. And because almost every animal on Earth already possesses the basic molecular ingredients, almost any animal is a potential candidate for venom evolution. And yes, even you have that same potential. A shrew, for example, is a small, frenetic mammal, seemingly unremarkable, but the short-tailed shrew of North America produces a venomous saliva capable of paralyzing prey several times its own size. The solenodon, an ancient, burrowing mammal found only in the Caribbean, produces venom through grooved teeth in a system that researchers confirmed evolved completely independently from shrew venom, even though both lineages recruited the same family of proteins to do it. There is a deeper reason why this keeps happening, and it comes down to an arms race that has been running for hundreds of millions of years. Life doesn't evolve in isolation. Every predator shapes its prey. Every prey shapes its predator. When a snake evolves venom potent enough to kill a mouse, that mouse's descendants face a new selection pressure. The ones that happen to carry proteins that slightly reduce the venom's effectiveness survive slightly more often. Then they pass that resistance on to the next generation, then the next. The prey becomes harder to kill. So the snake has to keep improving. This is called the co-evolutionary arms race, and it drives venom forward with a relentlessness that ordinary selection can't match. Honey badgers can shrug off cobra bites that would kill a large dog. Mongooses carry a mutated receptor that makes them largely immune to cobra neurotoxins. Certain ground squirrels have evolved blood proteins that neutralize rattlesnake venom before it can act. And each time prey builds resistance, the predator faces pressure to evolve a new chemical strategy. Different toxins, different targets. In 3 separate lineages of cobras, venom shifted function entirely, moving from a predatory tool to a defensive one, evolving the ability to spit venom at the eyes of large mammals. Each of those 3 lineages evolved this behavior independently. Now here is where it gets genuinely strange. Venom is not always the product of the animal carrying it. The blue-ringed octopus is one of the most venomous animals in the ocean, a creature the size of a golf ball capable of killing a human with a bite that is sometimes not even felt. Its venom contains tetrodotoxin, one of the most potent neurotoxins known, but the octopus doesn't produce it. Bacteria living inside its body do. The octopus has evolved a relationship with these microbes, housing them, concentrating their toxins, and deploying them as a weapon it never had to build itself. This is venom evolution at its most opportunistic. Rather than building a chemical system from the ground up, an animal finds a shortcut. It outsources the chemistry to organisms that have already solved the problem. Evolution, it turns out, doesn't care how you get there. The cone snail takes this to an almost absurd extreme. There are around 800 species of cone snails, each one slow, defenseless in a conventional sense, living on the ocean floor. But each species has evolved its own entirely unique chemical arsenal, not variations of the same venom. Distinct cocktails tailored to specific prey. Fish hunting cones produce one set of compounds. Worm hunting cones produce another. Mollusk hunting cones produce another still. Over 2,000 distinct conotoxin sequences have been documented so far, and that number keeps climbing. Every cone snail is running its own pharmaceutical company, experimenting constantly, generating new molecular variations, testing them against prey that is itself evolving resistance. In a single genus, evolution has produced more chemical diversity than most drug companies have ever synthesized in their entire existence, which is why scientists are paying very close attention. Captopril, one of the world's most widely prescribed blood pressure medications, was developed from a peptide found in Brazilian pit viper venom. It works by blocking the same biological pathway the snake evolved to exploit. A peptide that spent millions of years perfecting its ability to manipulate mammalian blood pressure has been redirected into saving human lives. Ziconotide, approved in 2004, is a painkiller derived from a cone snail toxin, and it is estimated to be 1,000 times more potent than morphine, without the risk of addiction. A compound evolved to paralyze fish now treats chronic pain in cancer patients who have exhausted every other option. Scorpion venom is being studied as a way to illuminate brain tumors during surgery, a compound that evolved to hunt insects, potentially guiding a surgeon's blade. These are not happy accidents. They are the direct result of hundreds of millions of years of biochemical refinement. Chemistry so precise, so targeted, that modern pharmacology is still catching up. So why does venom keep reinventing itself? Because the problem it solves never goes away. Every animal faces the same fundamental challenge that drove the first venomous creature to evolve its chemical arsenal. How do you catch something that doesn't want to be caught? How do you defend yourself against something that could kill you? How do you compete in a world where the line between predator and prey shifts constantly, unpredictably, across millions of years? Venom is evolution's most elegant answer. It turns ordinary proteins into weapons. It turns prey into statues. It turns a slow-moving snail into one of the most dangerous animals in the ocean, For every venom compound that has made it into a medicine, thousands more remain unstudied. We have been surrounded by the world's most sophisticated pharmaceutical library for hundreds of millions of years, and we are only just beginning to read it.